So today we will discuss the PIQ of 2025 for the genetic portion. Okay, the which came in the 2025 the plant science JRF 2025. Okay, in the first question, the which came from the genetic portion is the Griffith test, but Griffith transformation experiment. It is done in 1928. Okay, the following activities were performed. Arrange the activities the proper sequential order from the first to last. In the Griffith test, the what did by the Griffith is the first they introduced the RAP stain. First, you should know that which type of stain he used. He used the two stains. If one is the R type, one is the S type. This is the RAP type. Okay, RAP type stain. That is avirulent type, not virulent type. But S stain is the produce the smooth colony, smooth colony, and it is a virulent type. Virulent type. Okay, first I introduce the R stain into the mice. Okay, but the mice, all the after the injection of this R stain, all the mice are alive. Okay, not died. But after when the smooth stain enter into the mouse, okay, the all the mice are died. But under it is the first condition. This is the second condition. In the third condition, what did he killed all the smooth stain. First, he killed all the smooth stain and introduced into the mouse body. But all the mouse are alive. Okay. But on the fourth condition, what happened? He used the dead stain of S after the killed, or you can write the kill S stain. Okay. Killed S stain plus along with the R, means this is the live condition. Okay, these two those two inoculations, when they enter into the mouse will die. Then what is happened? He found that the some in the cell of the in the blood of the died mouse, he found the live live stent. It is called the transformation principle, and thus the fragment from the R stent the fragment of the A stent that cross the R stent to modify it into A stent, means the R stent modified into A stent by the fragment from the A stent. Okay, this is called the transforming principle. Transforming principle, you should remember. Okay, and it helps to transform the R to A stent. Okay, so the answer will be correct is the first is the R stent they included, then they use the R stent live, then first is, then second is, then after that he include this injected the heat kill S stent. Okay, then this done the infected mixture of the heat kill stent and the R stent. So what will be the correct answer? ACDB. Second answer is the correct answer. Okay. The second question is the select the rRNA, ribosomal RNA associated with the eukaryotic ribosome. In the eukaryotic ribosome, both prokaryotic and eukaryotic contain the two subunit. You always know. Okay, you know. This is a small subunit. This is a large subunit. In the small subunit, the prokaryote contain the 30 subunit, and it is called the 50S subunit. It contains 60S RNA only. Okay, but the large subunit contain the 23S small RNA and the 5S RNA. Okay, but in the eukaryotic, it is called small fragment of the ribosome is called a 40S, and large fragment of the ribosome is a 60 subunit. Okay, it's contain the 28S RNA, not the 23S. 23S it does not contain. Okay, so it will be not correct. Okay, but it contain the 5.8S, it is correct, and it is also 5S. Okay, rRNA, and it is also 18S rRNA. Okay, so it is also correct. So it will be incorrect, but fourth are correct, means the ACD is correct. ACD, The fourth option is correct. Okay. The inheritance of mitochondrial DNA in most sexually producing organisms exhibit a unique pattern of the primarily due to. You should know the mitochondrial DNA or mitochondria only transfer. It is a cytoplasm. Cytoplasm. This is the nucleus. This is the nucleus. Okay. This is the female. Female. Okay. In the male, it is the nucleus of the male, and this is the cytoplasm of the male parent. When this crossed among themselves, only the cytoplasm of the maternal parent come in the progeny, not come from this parent. From the male, the cytoplasm not come. Okay, so the mitochondria present in cytoplasm only, so the mitochondria only coming from the maternal parent. Okay, not coming from the paternal parent. Okay, so this is the correct answer, would be the maternal inheritance of the mitochondrial genome is the correct answer. Because the paternal type of the inheritance is generally not happen. Primarily if used, then this is the correct answer. If it use either mitochondrial DNA can be transferred, you can use the both parents. Sometimes the male from the male parent, the cytoplasm also transmitted to the progeny sometime, but most of the primarily if they use this primary word, you should use this correct this answer. Okay, maternal inheritance will be happen. Okay. Fourth option, the consider hypothetical repressible operon under negative control. A mutation in the gene encoding a repressor, you can say it is called the regulator gene. Okay, protein result in the non-functional repressor. Generally regulator gene produce a functional repressor. Okay, but if the mutation is happening, it should be the non-functional repressor that cannot bind with operator. What was the expected expression pattern of the structural gene in the operon? First, you should know the what is the contribution and important function, and how the repressor protein regulate the function of the operon. Okay, this is the operon structure. This is the promoter. From the promoter, the some portion is the operator region. This is the regulator segment. Okay, regulator gene. Okay, this is the promoter. This is the gene 1. This is the gene 1. It is the gene 2, and this is the gene 3. Okay, under normal condition is produced the active repressor. Okay, active repressor protein, it is bind with the operator segment and prevent the activity, prevent the binding of the RNA polymerase. Okay, prevent the RNA polymerase, so these genes are not transcribed. Okay, but if the repressor are non-functional, means if the repressor are non-functional, means this produce from this type non-functional protein of the repressor that cannot bind here. So every time RNA polymerase can bind to this promoter, and this produce the the transcription will be happen. So every time that constitutively the RNA transcription will be happen. So second, third option will be correct. Expression would occur constitutively regardless of the presence or absence of the co-repressor. And what is the function of the co-repressor? Co-repressor sometime bind with repressor, activate the repressor. After that bind with this operator region and the prevent the activity of the RNA polymerase. Okay, so third option is correct. In the typical diploid rice plant, the 2n is equal to 24. Which of the following could be the example of the aneuploid form? You all know that the aneuploid, if the 2n is the diploid parent, what the plant you can call the aneuploid. Aneuploid contain one or two or more, few, only few more chromosome, or the few less chromosome. These are called the aneuploidy, okay. But what is it called? Euploidy. Euploidy means it will be the individual contain the half, or the only whole genome will be multiply or the reduce from the individual. That is called the euploidy condition. Means the individual may be n, or the 3n, or the 4n. Then this condition are called the euploidy. But when the one or few chromosome included, added, or removed, or deleted, then this individual are called the aneuploid plant, or this condition is the aneuploidy. What is the type of the aneuploidy? This is the hyperploidy and the hypoploidy. Hyperploidy means adding of the one or more chromosome. Hypoploidy the deletion or the remove the one or more few chromosome. The different type of the hyperploidy is present. This is the trisomy. The only one chromosome is included. Double trisomy means two different chromosome adding. Okay. A tetrasomy means the total two homologous of a chromosome is included in the individual present. Okay, this is called a tetrasomy. A double tetrasomy means two different chromosomes. Two chromosomes, three chromosomes. The homologous chromosome of the 2 and 3 include here more amount. Okay, then this is called a double tetrasomy. What is the pentasomy? If the three of a particular chromosome included here, then this is called a pentasomy. What is a monosomy? Sometimes the monosomy question coming in the GRF, you should remember. And okay, in this condition the one chromosome is removed and not contain only if the 2n is equal to 24, the monosomy condition will be the 23. Okay, in the rice plant. Okay, if the 11th number chromosome only present the one, then it can be right that like the 2n minus 11th number chromosome. Okay, then this is called the monosomy. Okay, and what is the euploidy? Euploidy means if the content, I have already told that if the whole genome is increase or the decrease, then this is called the euploidy condition. This is the haploidy, diploidy, and the polyploidy condition. And polyploidy is two types: the autopolyploidy and allopolyploidy. If the AA genome multiply and this produce the AAA genome, this is called the autopolyploidy. If the AA and the BB genome merge with each other and produce AA capital B genome, then this is called allopolyploidy. Okay. 6 number question is a series of underbreaking experiments were conducted by various workers that collectively led to scientific understanding that DNA is a genetic material. If one term is coming, DNA is always, if the term the always genetic material, you should remember this is not correct. Sometimes RNA also may be the genetic material. Some viruses also. Okay. In the Hershey and Chase, the DNA as a genetic material they proved theoretically the transforming principle concept given. These are also related to the genetic material, and the transforming principles are actually DNA. This proved by the Avery, Magliot, and McCarthy. Okay, experiment 1944. Okay, but in the such Frankel and Cornard experiment is done in the tobacco crop, the tobacco mosaic virus, and they found that the tobacco mosaic virus contain the RNA as a genetic material. So the A, B, and third, all the scientists are related with the DNA as a genetic material. They proved, but in the Franklin and Cornard experiment done did that RNA as a genetic material. So the correct answer will be A, B, D. A, B, D, second option. Okay. Seventh question. The transformation is a natural process in which a bacteria can take up the naked DNA from the environment. You know that transformation, bacteria, if the DNA is present, DNA fragment present in the medium is the medium, they take and enter into this bacteria enter into the nucleoid. Okay, this is called the transformation. It is a correct statement. Transformation does not cause genetic recombination. It does cause the genetic recombination. Not, it is a correct. So statement is correct. Means the transformation cause the recombination. Transformation of bacteria. Transformation of bacteria cause the recombination. Okay. So third option is correct. Statement 1 is true. Statement 2 is false. 8 number question. The eukaryotic cell organelles and the major function. Here the generate most of the cell supply of adenosine triphosphate or ATP. You know ATP generally produced by mitochondria. So A will be the correct answer is A is equal to 3. Okay, A is 3. So among these two, one will be correct answer. Endoplasmic reticulum. Generally, I mean, if the endoplasmic reticulum generally produce the protein because the ribosome present on the endoplasmic reticulum. So this is called the RAP. So this is the site of protein synthesis. So B will be 1. So third option is correct because B is 4. So here the third option correct. And the Golgi apparatus produce a modified sort the package of protein. C option 4. C option 4. And the lysosome generally produce the digestive enzyme to break down the waste material and the debris. So D is equal to 2. So third option is correct. Okay. Likelihood of an individual in a population carrying two specific allele of a DNA marker, each of which has a frequency of 0.3, means this human DNA marker has the two DNA marker have two alleles, A2 and A1. Okay, and frequency of A1 both have 0.3. Frequency of A1 is 0.3 and frequency of A2 is 0.3 also. Okay, and what is the probability of the likelihood of an individual in a population carry two specific allele? Means one individual that carry both A1 and A2. We know if the A1 it is formula is a p square. When A1 A2 it means 2pq. Okay, mean this is the p and this is the q. So and the A2 means q square. So they want to know the what will be the probability of an individual of this type. Okay, so this will be probability of A1 genotype or the individual will be 2pq, means 2 into 0.3 into 0.3 means 2 into 0.09, so 0.18. So correct answer is the third option. Okay. 10 number question. The point of the term gene phenotype, this term given by the Johnson. Okay, and construction of the first accurate physical model of DNA. You know that DNA model given by the Watson and Crick. So A is the third. A is equal to third option. Okay, among these three. Okay, construction of first accurate physical model of DNA means Watson and Crick, means B is equal to 1. B is equal to 1. So 4 option is correct. A is 3 and B is 1. Okay, you can also check the C is 4, means the confirmation of the semi-conservative replication of DNA. You know the semi-conservative replication model given by the Maselson and the Stoll, and discovery of the Chargaff's rules given by the Chargaff. Okay, so the fourth option is correct. 11th number question. The restriction fragment length polymorphism, and this is called RAPD, and there's a amplified fragment length polymorphism, or RAPD means randomly amplified polymorphic DNA. This is the restriction fragment length polymorphism. You should remember this is the full form of this marker: simple sequence repeat, restriction fragment length polymorphism, random amplified DNA, amplified fragment length polymorphism, and simple sequence repeat. Okay, the tandem repeat amplified by specific flanking marker in SSR microsatellite region. A or T, AAT means the repeat region is present. Okay, this is called the simple sequence of the microsatellite region, and the SSR developed by the flanking region of the this repeat region. This is mean the tandem repeat amplified specific flanking marker. This is so the SSR is equal to the third option. So the D is third option. Okay, maybe this is, you can check also AFLP. In AFLP method, what is done in AFLP? The two type of restriction nucleus use. Endonuclease enzyme use. Okay, one is the rear cutter. This is the first type of restriction enzyme. A second is the rear cutter and the Fuquin cutter. Okay, the polymorphism detects a restriction enzyme and southern blot. This is mean this RFLP. In RFLP, only one restriction enzyme cut. Only restriction one enzyme is used. EcoR1. Okay, and after the restriction happened by the restriction enzyme, this fragment are moved in the southern blot. After that, your probe is utilized and checked is present or not. Okay, this is the done in the this or means the A is equal to 2. Okay, in AFLP, what is done? In AFLP, the combined restriction digestion means the two type of restriction digestion is done, and after that the PCR is done. What is the difference between AFLP and RFLP? In RFLP, after the restriction enzyme digestion, the southern blotting is done, but in this condition, the AFLP method, after the restriction digestion, the PCR is done. You can also know the site specific PCR amplification. Okay, is done, and the RFID, what is done? Only random marker up to 10 or 20 nucleotide marker, random marker used. Okay, and after that the PCR is done. This is the use of the short arbitrary primer. Arbitrary means the random primer is used under the low stringency PCR. Means low stringency means temperature will be low and other stringency conditions should be reduced so that this can be bind with the DNA. Okay, so the what is the correct answer? RFSB is equal to the this D is equal to 3. D is equal to 3 and RFPD is equal to restriction RFLP is equal to 2 means A is equal to 2 and B is equal to 4 means what? RFPD means IFLP C is equal to 4. Only one, two option is theirs. Maybe this, maybe this. Here's what is happened. The SSR, so B is equal to B1 or 2. B means RAPD. B means RAPD. This is the 2. So third option is correct. Okay. Means RFLP A is equal to 1. Sorry. A will be 2. This is the A2. B means RAPD. RAPD means B is equal to 1 should be. Okay. Means this is the correct answer. A is equal to 2, B is equal to 1, C is equal to 4, D is equal to 3. You can also check again. RFLP A option 2 means the polymorphism detection restriction southern. Then after the southern plot is done. Okay, so A is equal to is correct. Then B is equal to 1. RAPD. RAPD means arbitrary payment is also correct. C is 4 means AFLP is combined restriction. C is 4. So D is equal to third option. Okay, third option. So first option is correct. 12 number question. The which of the following base describe the primary mechanism by which the histone modification contributes to heritable changes in gene activity without altering the DNA sequence? Means the histone modification generally does not change the base sequence. Base sequence does not change. Okay. only change the histone. Maybe it is a methylation or the acetylation. You should remember, if the histone modification done by acetylation, it helps to increase the expression expression of DNA or gene. Okay, if the acetylation happen, okay, and methylation, if the histone modification is done, then in this condition maybe gene expression increase, maybe decrease. Okay, so this is the correct answer will be that they influence the chromatin compaction and accessibility to transcription factor, because in acetylation it's help to reduce the compactness, okay, and transcription factor is bind under. If the chromatin compaction increase, this condition happen by the methylation, means methylation increase the compactness and acetylation reduce the compactness and increase the accessibility to transcription factor to increase the expression of the gene. Okay, so third option is correct. In the classical operand structure, which consists of a cluster of gene under a control of a single promoter, generally operon controlled by a single promoter, is transcribed together into the single polycystonic mRNA. It is correct, because if the operon is there, the number of gene number 1, 2, 3 is there, this is the promoter, then they together transcribe. Okay, they together transcribe. After they remove from each other, they produce 3 RNA. Okay, after the processing, actually this is produce the polycystonic RNA. This is called polycystonic RNA, because cystone means the 3 cystone present. Okay, so this is called a polycystonic. This is primarily found in the prokaryote, but eukaryote sometimes the less number of polycystonic mRNA is found. Okay, the most of the time in the eukaryotic cell, the most of the mRNA are monocytonic in nature. Okay, so correct answer will be the third. Okay, the respiratory chain of bacteria is associated with the cytoplasmic membrane. Okay, this is the correct, because the ETS cycle, electron transfer system cycle, generally happen in the respiratory. Also, you can tell the respiratory chain of bacteria associated with the cytoplasmic membrane. And statement 2 is the respiratory chain of the eukaryote present in the nuclear membrane. It is incorrect, because in eukaryotic cell, the respiratory by ETS chain present in the inner membrane, inner membrane of the mitochondria. Okay, so the correct answer is the statement 1 is true, statement 2 is false. So third option is correct. 15 number question. In replication of DNA, the leading strand is synthesized continuously in the same direction. Okay, you should remember same direction as the movement of replication frog. Require only the one initial primer. Here the important term is the one initial primer is required. The movement is the same direction along with the movement of replication frog. Okay, and this is the leading strand. Okay, so statement 1 is correct. And in statement 2, DNA replication, the lagging strand is synthesized continuously. Okay, it is also correct. Here the continuously happen, but in the lagging strand, it is discontinuously happen. Okay, in opposite direction of the movement of replication frog. In the short fragment called the Okazaki fragment. Okay, this is called the Okazaki fragment, and each Okazaki fragment requires a new RNA primer. Is also correct. So both are correct. So answer will be 1. You should remember first that this is the double stranded DNA. After this is the ORC region. In the ORC region, the proteins bind with this ORC region. After this helicase coming, and the two strands separate and from the fork, means the helicase enzyme moved this direction also and this direction also, and this is the fork direction, this is the second fork direction, and this is called fork region. Okay, two fork produce. Okay, I can't explain in the another slide. Okay. This is the 3 to 5 prime direction. This is the 3 to 5 prime direction. This is the 5 to 3 prime direction. Okay. In the 3 to 5 prime direction, the it is called the leading stand. Leading stand. Okay. This is called lagging stand. Okay, lagging stand. In the leading stand, what happened? Only one primer is used. Only one primer. Only one primer. Only one primer is used. Okay. This is the 5 prime direction. This is the 3 prime direction. Okay. And the DNA from, wait, DNA from continuously. Continuously. This is the 3 prime direction. This is the continuously from. Okay. But this is called lagging stand. What happened? In lagging stand, the different multiple number of the primers, RNA primers is needed. Okay. And here's what happened. The synthesized fragment is very short. Synthesized fragment is very short. And this is called the Okazaki fragment. Okazaki fragment. Okay. Okazaki fragment 1, 2, Okazaki fragment 3. Number of Okazaki fragment is there. After that, they ligate each other. After this is removal of this RNA and the bind. This is done by DNA polymerase 1. DNA polymerase 1. Okay. And this is the discontinuously, this is the discontinuously, this is the continuously. And what is the direction? Is the direction of the frog direction here. This is the direction, the same direction, but in the lagging stand the DNA polymerization happen in opposite direction. Okay. So the correct answer is the broth. Okay, is correct. What is the second 16 number question? A cross between two breeding lines, one with a dark red flower, one with a bright white flowers. Means parent one is the red flower. Okay, red in nature, red flower. Okay, and the parent two is a smaller white flower. Okay, when they cross them, they produce capital or smaller. Means capital or smaller. This is the F1. But when this is the intermediate in nature. Intermediate means intermediate red color. Intermediate red color in nature. When this helping is done, okay, helping is done. This produce capital or smaller and smaller. Okay, this is for 1:2:1. Okay, this is the intermediate red color, this is the red color, and this is the white color. This means the 1:2 ratio. Dark red, this is the dark red, and this is the light red, this is the white. Okay, but you can sell that the intermediate red also. Okay, this is associated with the incomplete dominance. In with complete dominance here, then it's produced 3:1. 3 is the dark red, and 1 is the white. Okay, plant. If the codominance is here, it's produce the dark red plant, and another plant contain both dark and the white, and the third contain only white plant. If the overt dominance is theirs, mean capital are smaller and capital. This is for your dark red. It is produce a very dark red. Redness will be more than this parent one. Okay, and the others will be smaller white in nature. So second option is correct. 70 number question. The four statement about the mitosis, identify the correct statement. The homologous chromosome pairing and synapses in the prophase 1 die critically mediated by the synaptomal complex. Homologous chromosome pairs and prophase 1 of the meiosis 1, it is correct. Okay, in pachytene stage. Means in zygote stage, the synaptomal complex formed. After that, the crossing what happened. Mean this is the zygote stage. Okay, In reductional division of the meiosis 1 followed by equational division in meiosis 2, this portion is correct. Okay, but where the sister chromatid separate reduce the chromosome number twice, it is incorrect. Only reduction is happen only one time. So second option is correct. B option is incorrect. First option is correct. Meiosis maintain the specific chromosome number species. It is correct because it is a maintained the chromosome number generation after generation. Okay, different progenies. Okay, by meiosis. Okay, this is called. It is also correct. And meiosis occur in reproductive cell because it is not present in the somatic cell, only present in reproductive cell. It is also correct. So A, C, D is correct. A, C. Third option is correct. Okay. Which of the following is not an assumption of the Hardy-Weinberg law in equilibrium? In the final answer sheet of the 2025, the plant science, the given that answer is the small population size is the correct answer. Okay, but you should remember the what are the assumption. What are the assumption? Not a assumption. It's a small population because the assumption is a large. It should be large population. So what is the assumption of the Hardy-Weinberg law? No mutation. No mutation. Okay, large population should be large in nature. Okay, population should be large. No mutation, no selection, no migration. Means all the natural factors should be not be present. Okay, should not be present here. The Hardy-Weinberg population. Okay, and the population should be large enough. So not the assumption is the third option is correct. Okay. 90 number question. The four major activities during replication in the prokaryote, arrange them in the sequential order, occurrence from the start to finish. The primary primase synthase the short RNA primers on the broad template stand provide the three ways of the start synthesis. The enzyme helicase further unwind the DNA replication from the separate the two strand. I already told that in the Odyssey region, the initiator protein bind to specific sequence. This is called Odyssey. This is called the initiator protein bind to the first. I have already told. Okay, after the helicase function happens. So this is will be the first. This is the second. Okay, after that the primase enzyme will be produce the RNA primer. So after that this happen, and that after that the DNA polymer is coming and produce the RNA DNA replication or add the deoxyribonucleotide. Okay, so what will be the correct answer? The D first, after that B, then the A, after that the C. So DBAC, DBAC. So the third option is correct. Okay. 20 number question, the find out the event or factor that are exception of Mendelian principle. In the Mendelian principle, only suitable only under the condition of the complete dominance. Okay, suitable, it is suitable. Okay, are what are the exception? Under exception, this is the all are coming incomplete dominance, codominance, and the polygenic state. This is the all are coming in the condition of the exception condition. So the B, C, D is correct answer. So fourth option is correct. Okay. 21 number question, in the context of the ex situ conservation of the plant genetic resource, what is the primary benefit of the integration of the molecular marker technology? First, the two type of conservation is there. What is the in situ type? It is conserve the material under here own area. Okay, but under ex situ condition, it is the from their area they are harvested or the removed from them and the conserved under different condition, the control condition. This is called the ex situ condition. And if the ex situ condition, maybe the seed bank, gene bank is there. Okay, in ex situ conservation, if they include the technology of the marker, it's help to efficient evolution of the genetic diversity and help to identify genetically redundant expression. Okay, so the second option is correct. At this fourth option, why it is not correct? It reduce the cost. No? This is the increase the cost and labor. So this is not correct. It ensure the concept jump and completely free from the all the harmful genetic mutation. It is also incorrect. Okay. They entirely eliminate the need of field-based phenotypic assessment. This is also incorrect. Along with the marker technology, the field experiment you should do. Okay. Then 22 number question, the AFLP amplified fragment length polymorphism combine the restriction enzyme direction with a selective PCR amplification gene. I already told that the ALP include the two digest enzyme. After the digestion, restriction digestion, they done the selective PCR. Okay, first P selective, after the selective PCR done. Okay, this is the correct. Second option, the AFLP typically function as codominant marker. You should remember this is the AFLP. AFLP generally it is a dominant in nature, dominant marker, dominant marker. Okay, it's can identified by any individual content or not any fragment by presence or absence, okay, this is the dominant in nature, not a codominant marker. Okay, so statement 1 is correct, statement 2 is incorrect. Okay, 23 number question, few statement about the crossing over genetic recombination and the recombination frequency. Choose the combination of the correct statement. First is the क्या बोला है, crossing over take place in the packaging stage. I have already told that the first the synaptic complex is done in the zygoting stage. After this crossing over done, okay, in the meiotic perpetuum, it is correct. Unequal exchange of chromosome segment occur between unequal, this is the not the unequal, this is the equal exchange. Okay, not the unequal, it is a incorrect in nature. Mean the equal exchange of chromosome segment occur generally of the non-sister chromatid of the homologous chromosome. Recombination frequency depend on the distance between the pair of gene. If the gene 1 present here and the gene 2 present here, if the distance is increased, if the present here, the distance increase, the recombination frequency will be increased. Okay, it is the correct. Recombination frequency generally does not grow beyond 50%. It is also correct. Okay, you know the why the does not exceed the 50%. It is the average. Okay, it is the average. You should remember this is the average value. Okay, behind which the recombination frequency cannot exceed. Okay, the gene located on the different chromosome exhibit independent assortment. If the chromosome 1 capital A, this is the chromosome 1, and the chromosome 2 contain B, chromosome 2. Okay, they does not dependent on B gene also does not dependent on A during the segregation. Okay, this is called independent assortment. So the B, C, D is correct answer. So fourth option is correct. Okay, the two genes G and H that are located on the same chromosome have the recombination frequency 20%, and individual is the G, capital G, small g, capital H, small h, is a cis configure, not a trans configure. You can also call it is a coupling phase. Coupling phase condition. Okay, cis configure, trans configuration is the call repulsion phase. Okay, repulsion phase. Okay, means it is a capital H, it is a capital G, it is a capital H. Here's contain the small g, and this content the small h. If the recombination happened, and generally parental type will produce two gamete, capital H, capital G, capital E. This is the parental gamete 1. Small ga, this is the parental gamete 2. A recombinant type will be capital G, small h. This is the recombinant gamete 1. recombinant gamete 2 is the small g capital H. Okay, this is the recombinant type. And these together form is the 20%, means the distance is 20 centimorgan. This together is the 20%. So these together will be 80%, because the 100 minus 20 is equal to 80%. So third option is correct. Okay, 80% parental type gamete and the 20% recombinant type. Okay. In the chi-square test with the two phenotypic class in plant height, what with the degree of freedom? You should remember in chi-square test, if the number of the phenotypic class is n, then degree of freedom will be n minus 1. Here the two phenotypic class is used 2 minus 1, so will be 1. 1 is the correct, 2 option is the correct answer. Okay.